All right, thank you. It's nice to see Noah. We overlapped at Caltech for briefly a month back then when we were working on feedback control for biology. All right, it's my pleasure to be here, and I started my lab in 2023, and then it's my pleasure to share the latest work that came out of my lab. Okay, so I'm in the chemical engineering department. This is how I convince people that we are doing something relevant to chemical engineering. So we're essentially in synthetic biology trying to engineer living cells and convert them to a little chemical process plant. And so all of the systems, including sensors, actuators, and the process and the control and the library are all necessary in our systems design. As a synthetic biologist, I am part of the movement, and we promised the world that we're going to evolutionize society by implementing synthetic biology to sustainable agriculture, sustainable material, bioremediation, and living therapeutics. But biology is not like any of the systems that we have ever engineered. So it is actually difficult, and it's especially important that we implement feedback control in it. In our lab, and the overall goal is to achieve autonomous control in synthetic biology. What I meant is that we want to engineer a bug where it goes to the environment and sends the signal where it needs to remediate. And once it senses that, not only it will produce the molecule to remediate that signal, it also scale up. And also, and when the signal goes away, it should know to scale back in population so we minimize the environmental impact of the synthetic bugs. So there are a bunch of questions that we need to answer. So one is that what chassis do we engineer? Is E. coli good enough for everything? How do we sense these targets? And what type of sensors we need to, what type of sensors do we need to engineer? And then also, how do we actuate both growth and production in these two interval scales? And finally, what type of feedback control to implement and how to implement them. All right, so let's go back to the central dogma of biology. So in synthetic biology, we basically look at this information flow and try to engineer and utilize it. So it's easy for us to write them into differential equations. So if we have a promoter that's inducible by X and is driving a GFP, and then we will be able to see that sorry, is there a chat? Okay. So the GFP, and then we were able to produce an mRNA, and the mRNA gets converted to a protein through translation. No, sorry, not converted, translated. Okay, and then now we can describe this process with two differential equations. It's very easy. You have a transcription rate, and then an mRNA degradation rate, and translation rate, and then in a protein division rate. And then you just have two species, M and P. And when I was in grad school, I learned that if you put a SSRA tag into your gene for the YFP, it will get tagged for degradation. So what it means is that this YFP now is marked as misfolded. So the intrinsic protease would attack it, so they will have an enhanced degradation rate. So it's exactly the same two equations. It's just the fact that this dp will get increased. So you have an increased degradation, and if you simulate these two systems, you're going to get something that looks like that. If you don't have the SSRA tag, the system will reach steady state a lot slower than the type you will have the SRA tag. And then this is expected. But over the years, when I actually did the experiment, this is what I get. There was never really a speed up, at least when I observe. And then there's sometimes by engineering the circuit, I can get rid of this oscillation. But there are enough variants of these type of circuit behave this way make me think that this is not a fluke. And also the same construct, if you repeat it multiple times with multiple replication, this is the dynamics you will get. And over the years, I just kind of swept under the rug, and I either don't use this, or I use it, and then I use a weaker SRA tag or mutate RBS or the promoter, just like what Noah did, and then to get rid of that. So what really happened, I try to understand that is when I looked at these equations, you see that we're assuming that the production rate, degradation rate, translation rate, and the dilution rate of protein are just constant. So that means that over time, these rates never change. And this is actually only true if the system stays in the log phase indefinitely. So in other words, this captures the single scale molecular dynamics when growth is constant, which is only true if the experiment is performed in a chemostat. And this is a chemostat. What happens is that you have a fresh feed that goes into the tank and your cell growing here, and you constantly pump out the content. But if you're a synthetic biologist, most of the time you're running the experiment in a well plate. And in a well plate, this is what happens. You're going to have the cells grow and through the three different stages. And then this can be described by a logistic growth curve. And then when cells are going through these stages and all the transcription and translational machinery are actually going to change. So none of these parameters are going to stay constant. So if we want to predict any dynamics or use a model to inform us how to implement feedback control, then a dual scale dynamical model is needed to capture system dynamics with any non-constant growth. So my student, Harry, he built a CRN model, so that stands for chemical reaction network. So this is a model that contains 42 parameters and 28 equations that captures every single detailed interaction and that happens inside of the cells in order for a DNA gets transcribed and translated into a protein, and we see them. And so, and all of there are 5 rate modification functions we came up with, and we use these RMFs to link gene insertion dynamics to growth. So for instance, if you see this blue box, and this means that translation elongation rate will go down over time when the cell reaches stationary phase. So this is a CRN, just basically a large version of the gene expression across growth stages model, and we call it the Geeks CRN model. So how does it do? It looks like it's able to capture the double peak dynamics, and apparently the model that links all the growth dynamics with the molecular dynamics and in dual scale is able to explain why I see this double peak. But we don't understand why. So because when we look at these 3 figures, this is the without the SRA tag, this is with SRA tag you saw earlier. This is the growth dynamics. And the interesting dynamics, actually the oscillation happens during the log phase. So we did a sensitivity analysis, and then we perturbed every single parameter of this model over time, and then to see how much impact it's going to make for the YFP signal. And we found that in this region where the double peak is happening, there are 4 parameters that stand out. That actually, this is the amino acid recycle rate and amino acid replenish rate. And then these 2 are the logistic growth rate. So we then took these 4 parameters and then we checked them on the phase diagram. So basically this phase diagram tells us how many peaks are there in this dynamics. So if you look at this in the middle, this is rate. When you have a trough or a peak or a minimum, then the phase diagram will cross this line. So this is going to be our highest peak, and this is the second highest peak, and then the lower one is the trough. So, but outside of this zero origin, if it crosses this line 3 times, that means we have this dynamics. And then we show that if we vary in Cmax, which is how much cell you're allowed to grow inside of the well plate, this double peak dynamics could disappear at one point. And the same things for the varying the logistic growth rate. And if we look at the amino acid replenish rate, this is also going to determine if we have a double peak or not. But if we vary in the amino acid recycle rate, it's not going to have that much impact, but it will influence how high the second peak is. All right, so that was a lot of equation, a lot of models. We don't really want to use that. So we try to reduce the geeks model. And so we wrote a reduced version. Other than this growth phase, this growth dynamics, there are basically only 6 equations in this version where all the bolded parameters are linked with growth. So it is also dual scale, but it's a lot more manageable. And then we're hoping that these type of modeling technique can be used to inform how to design feedback controls. And then we found that, so the red is the effective and the one we built here, and the CRM model is the ginormous one I showed you earlier. And there are 2 of them in between. So we see that at the very least for these type of oscillatory dynamics, it is important to have a CRM model. So there is a trade-off between the accuracy and the model size. But with that in mind, can we use the effective Gates model to capture closed loop system dynamics with non-constant growth? So if we were to use the model to predict how the dynamics looks like if we want to implement a feedback control in the well play, is it going to be accurate? So the next part is we're trying to use optogenetic control and to make this type of feedback loop. So the idea is that if we treat the cell as a process and I use LED light as the actuator to control gene expression, and then have a piece of electronics to sense the cell gene expression, and then also compute as a controller, and then link to the actuator, can we actually get this loop to link up, and how does that work? So the first thing we wanted to characterize is this optogenetic control that came out of the Tabor lab at Rice University. So what you do is basically engineer the cell so that when you have a green light that shines onto the cell, it will make GFP. If you use a red light shine on the cell, it will not make GFP. So this experiment, because it requires a constant light source, so what you need to do is just put it in like a cardboard box with a LED light tape on top, and sort of throw everything into an incubator. And this is what I did when I had to do this manually. So basically every hour my alarm just goes off and I go take the plate out of the light box and I put it in the plate reader and measure the dynamics. But I don't want to do that. So we need a prototyping device that can automate this measurement. So back at Caltech, I had this really brilliant undergrad, Krishna. He built me one of these. So this is the plate he built. So this is the outer shell. That's basically commercially available, just the plate, well play shell we use. And then he 3D printed this frame, that half of it contains all of the electronics. There's a microcontroller, battery, and then he has 16 LED lights inserted inside of in between these 2 rows. And then every time you run an experiment, you just print out a PDMS stick film and then you drop it in there, and then you run the experiment, and then these can be sterilized, and after the experiment, you can throw it away. And we were able to run the experiment. And if we use the effective model that's not linked to growth, this is the dynamics you will see. And if you use the effective model, we were able to capture it much better. And then the idea is that we're going to put this plate into this plate reader, and the signal gets read by Gen 5. Gen 5 tells there's a Python code that every time it updates and Python code reads it, and then it tells Arduino. Arduino links with the plate, and then they adjust the LED light. So the first thing we did was to do a proportional control, and we set two set points. One is the purple one is the orange one, and then we saw that consistently it misses the set point and has a offset. And this is actually captured by our model as well. So if we just use our Geeks model, the Geeks effective model, and to describe this process, we saw that this overshoot slash offset is captured. And so we wanted to understand why is that. So we then used the highly reduced version, so the three equations, but all of these parameters are still growth dependent, and we're able to simulate system where there's no offset. And then we use the model and then to see how long is the dead time when over time when we want to switch between light signals, how long does it take for the system to respond at all? And then we plot the dead time, and then use the dead time. Then we told the system, the controller, to measure the signal in the previous time, that basically subtract the dead time. Then we're able to use this model to simulate this overshoot and this offset. So essentially, this means that this offset is caused by the dead time that is intrinsically existing in the biological process. And we were able to use the model to see that if we were to implement an integral control in there with this, this is not anything biological, just computed inside of the code, and it should be able to eliminate the offset for the higher set point. But for this one, it's still going to be overshooting. And then if we to put a D control in there, it should be able to eliminate that. We're very excited about the results, and hopefully we can do the experiments to verify that. All right, so right now my group, there's half of the group is doing safety and autonomous control in living therapeutics. So we're engineering a non-model organism in the lab, and then apply autonomous control we learned using the model and mathematical equation to achieve safe and reliable bacterial living therapeutics for cancer. All right, so I want to thank everybody in my group and my collaborator, Krishna. He graduated from Caltech and now is a first-year PhD student at MIT. And then my former colleague and now collaborator, Yush Pandey, now is at UC Merced as assistant professor. And my funding source is our page.